Students, today we will discuss about the botany, cultivation and economic importance of sugarcane. Sugarcane, family Poaceae, genus Saccharum, species Opsinarum, diploid chromosome number 2n equal 80 and basic chromosome number X equal 10. Genus Saccharum comprises of 6 species, namely Saccharum Opsinarum, Saccharum spontaneum, Saccharum robustum, Saccharum eduole, Saccharum barberi and Saccharum sinensis. Saccharum opsinarum, also called noble cane. This species is derived from Saccharum robustum. It is a soft, thick-stemmed, large-barreled with a low fiber and high sucrose content. Diploid chromosome number 2n equal 80, but in some varieties it is 81 and 98, 99. Saccharum spontaneum. This species is smaller, highly polymorphic, disease resistant and highly vigorous, having diploid chromosome number 40 to 128. Saccharum barberi. This species is thin-stalked, suited to semi-tropical and temperate areas, thought to have arisen in North India as a hybrid of Saccharum spontaneum and Saccharum opsinarum. Saccharum sinensis. This is tall, vigorous, hardy cane. This species is thought to have arisen as a hybrid of Saccharum spontaneum and Saccharum opsinarum. Saccharum robustum. This species includes a group of canes which are indigenous to New Guinea. They possess mostly thick stalks with a relatively low sucrose content. The chromosome number is usually 80 but varies from 60 to 194. Saccharum eduole. This is morphologically similar to Saccharum robustum, except that the flower spike or inflorescence is compacted. Saccharum eduole is thought to be mutant form of Saccharum opsinarum. Sugarcane is grown in all tropical and subtropical regions of the world, on both sides of the equator up to approximately 35° north and 35° south. The most favorable temperature for subtropical and tropical cultivars is 25 to 26° centigrade. Optimum temperature for sprouting of stem cuttings is 32 to 38° centigrade. Tropical cultivars grow noticeably slower at 21° centigrade and at 13° centigrade don't grow at all. The water requirement is high. In moist growing areas, the cane needs 1500 to 1800 mm rain. In 2007, the main sugarcane producing countries were Brazil 33% of world's production, India 23%, China 7%, Thailand 4%, Pakistan 4%, Mexico 3%, Colombia 3%, Australia 2%, the United States 2% and the Philippines 2%. India. In India, Uttar Pradesh and Bihar are by far the leading states in growing sugarcane. It is grown chiefly in South India, especially Coimbatore and Punjab. In India, the area under sugarcane cultivation and total yield are 4.63 million hectares and 322.94 million tons respectively. Botanical description. Family Poaceae, genus Saccharum, species Opsinarum. Grows 8 to 20 feet tall and 2 inches thick. The main parts of the sugarcane plant are the stalk, leaf and the root system. Stalk. Sugarcane stalk is roughly cylindrical. The stalk consists of segments called joints. Each joint is made up of a node and an internode. The length and diameter of joints vary widely with different varieties and growing conditions. In general, however, the joints at the base are short and internodal length gradually increases. A cross section of an internode shows from the outside to in center the following tissues: epidermis, cortex or end, and ground tissue with embedded vestibular bundles. The epidermis is a single superficial layer of cells that exhibit different patterns which are variety dependent. Generally, the patterns are formed by two cell types, the so-called long cells and short cells. that alternate with one another. The cortex consists of several layers of cells just inside the epidermis. The cells of the cortex are thick-walled and lignified. These cells help in strengthening the stalk. More towards the center, the ground tissue contains the vascular bundles with xylem and phloem. The vascular bundles are much smaller and more prevalent toward the periphery of the stalk. Xylem tissue conducts the water and dissolved minerals upward from the roots, and phloem conductive tissue transports plant manufactured nutrients and products for the most part downward towards the roots. The vascular bundles are much smaller and more prevalent towards the periphery of the stalk. Xylem tissue conducts water and dissolved minerals upward from the roots, and phloem conductive tissue transports plant manufactured nutrients and products for the most part downward towards the roots. Leaf. Leaf of sugarcane consists of a sheath and the blade separated by blade joint. The sheath, as its name implies, completely sheaths the stalk, extending over at least one complete internode. A cross section through the leaf blade shows three principal tissues: epidermis, mesophyll, veins or fibrovascular bundles. The epidermal cells protect the underlying tissues from injury and drying. The mesophyll or middle leaf tissue contains the cells that perform photosynthesis. The fibrovascular bundles contain the xylem and phloem elements, which conduct water and nutrients to and from the leaves. In addition to the already mentioned tissues, there can be found other fibrous tissue for aiding in shaping and mechanically strengthening the leaves. The blade joint is where two wedge-shaped areas called develops are found. The ligule is a membranous appendage inside of the sheath, which separates the sheath from the leaf blade. It is a slightly asymmetric organ whose color, size and shape are age and variety dependent. The auricles, as their name implies, are ear-shaped appendages located at the upper part of the sheath margin. Not all sheath margins have auricles. Leaf pubescence, or the covering of various leaf parts with short hairs, is also variety and age dependent. Pubescence is not found on the leaf blade of commercial varieties, but does exist in sugarcane germplasm. Sheath pubescence can be used to identify plants. Root system. The root system is fibrous. Two kinds of roots develop from a planted seed piece. The set roots, which arise from the root band, are thin and highly branched. The shoot roots, originating from the lower root bands of the shoots, are thick, fleshy and less branched. A longitudinal section of a root tube consists mainly of four parts: the root cap, the growing point, the region of elongation and the region of root hairs. The root cap protects the tender tissues of the growing point as the root pushes through the soil. Now, the second, growing point, consists mainly of an apical meristem where cell division takes place. Third, the region of elongation. The cells increase in size and diameter until they reach their ultimate size. Fourth, the region of root hairs is characterized by epidermal cells forming outgrowths, that is hairs, which dramatically increases the root absorbing surface. Inflorescence. When a sugarcane plant has reached a relatively mature stage of development, its growing point may, under certain photoperiod and soil moisture conditions, change from the vegetative to reproductive stage. This means the growing point ceases from forming leaf primordia and starts the production of an inflorescence. The light silvery tan colored inflorescence, commonly referred to as tassel or an arrow, is an open feathery or woolly panicle about 0.3 to 0.6 meter in length and is produced only in paired spikelets, the lower being sessile while the upper spikelet is stalked. Both spikelets are surrounded at the base by a ring of long silky hairs, which impart a characteristic silky appearance to the inflorescence. Cultivation. The cane is vegetatively propagated using stem cuttings, usually with 2 nodes. The best material for cutting comes from 8 to 9 months old unripened canes, preferably from one's own propagation nurseries, with strict phytosanitary measures. 1 hectare gives enough cuttings for 5 or 6 hectares. Before planting, the cutting should be treated with fungicides and insecticides. Frequently, a hot water treatment 1.5 hours at 52 degrees centigrade is also used to kill viruses and insects. The row spacing depends on the cultivar and also on the width of the implements which will be used. It is normally 1.2 to 1.5 meter. The pieces of cane are laid end to end in furrows, either by hand or mechanically, and then covered with 3.5 centimeter soil. Roots are quickly formed at the nodes, then the eyes sprout, and after only a few weeks a permanent root system is formed. Until the closure of the leaf canopy, the weeds must be kept under control, either mechanically or chemically. It is most efficient to use herbicides only within the plant row and to use mechanical methods to keep rest of the space between the rows free of weeds. Even on good soils, top dressings of nitrogenous fertilizer, mostly urea, are necessary on oxygen-enriched soils, as well as an application of KCl. Sugarcane is better suited than any other plant for being fed with exact dosages of fertilizer. Analysis of samples from the leaves or internodes allows a calculated control of fertilizer requirement. Sugarcane is self-compatible and is sometimes grown in monoculture for many years without a break. In cooler subtropical regions, the vegetative period ends with the onset of cool season. There, the cane is freshly planted every year. In the tropics and warmer parts of the subtropics, growth begins again after harvesting. Under favorable conditions, one field can be harvested up to 8 times. Uncontrolled fires can cause major losses in sugarcane. Obvious precautions are prevention of fires and cultivation measures to prevent their spread. Constant weeding is essential since sugarcane cannot withstand competition from weeds. Harvesting. Depending on the climatic and growing conditions, the cane is harvested between 9 months and 2 years after planting, and in some cases even longer. The best time for cane cutting depends on the sugar content. With canes that have been already growing for a year, new canes grow in a second year, and the average sugar content must be calculated from all the stems of different ages. In humid tropical areas, where ripening is not determined by cool or dry weather, the entire crop can be made to ripen at a chosen time by the use of a chemical treatment. Cane is still often harvested by hand. The stems are cut off near the ground with a heavy knife, the green tops are cut off and the leaf sheaths stripped off. The work is made easier by burning the fields before the harvesting. In large plantations, heavy machines spread up the harvest. They lop off the tops, cut the canes near the ground, or uproot them, and sometimes also chop the cane into smaller pieces. Sustainable harvesting machines have also been developed for small plots of land and for sloping land. Processing should take place as soon as possible after cutting. In general, within 48 hours, otherwise heavy losses can be suffered. Mechanically harvested cane must be cleansed before sugar extraction. In the traditional method, after chopping, the cane passes through 4 to 6 roller mills in order to separate the juice from the fiber. For some years now, the diffusion techniques which were developed for sugar beet have also been used for cane, preferably in combination with roller or screw press. The crystallized and centrifuged sugar is then produced by clarifying, evaporating, fractionally crystallizing and centrifuging of the mother liquid. Sugarcane breeding. Breeding is a process of evolving new genotypes which are superior over the existing ones for the set of different traits of sugarcane. Modern sugarcane cultivars are the products of crosses between species of genus Saccharum that were made by breeders in late 19th century. The most important species contributing to modern sugarcane varieties were Saccharum obscinarum, commonly known as noble cane, which were widely cultivated for its ability to accumulate sucrose in its stalks, and Saccharum spontaneum, which is a vigorous, widely adapted wild species. The sugarcane breeding played a vital role in improving sugarcane yield and sugar recovery. Sugarcane Breeding Institute, Coimbatore, India, one of the pioneering sugarcane breeding institutes in the world, was established in 1912. The main objective of evolving superior varieties of sugarcane suitable for various agroclimatic zones in India. Breeding goals are to improve weight of the cane's sugar content, juice purity, and climatic adaptation. Disease resistant, especially against viruses. Adaptation to the photo period, non-flowering. Good tillering and good ability to regrow. Upright growth and leaves that drop when old. High yielding varieties. Some of the present grown varieties that have contributed most to the production of sugarcane in different parts of the world are Tucumán varieties Argentina, POJ 2878 and ML 318 Cuba, H37 1933, H32 8654, H32 1063 Hawaii, CO 421 and CO 419, CO 205, CO 312, CO 453, CO 740 and CO 421, CO 205, CO 312, CO 453, CO 740 and CO 331, Coimbatore, India. Economic importance of sugarcane. Sugarcane is globally an important source of commercial sugar, accounting for nearly 70% of the world's sugar production. Sugarcane is an established agricultural field crop with long history of safe use. Sugarcane is the most important cash crop of our country, from which sugar sucrose is produced in the factory. Sugar, gur or molasses are also prepared from sugarcane juice by evaporation of water contents. Sugarcane is primarily grown as a source of sugar. Sugar is sweet crystalline substance and is important source of sugar in human diet. Enormous quantities of sugar are used in the manufacture of alcoholic beverages, soft drinks, confectionery, ice creams, chocolates. Cereal manufacturers use sugar as sweetening agent, as do many other food industries. It also goes into hair tonics, explosives, photographic supplies, and medicines. It helps in the tanning of leather, the silvering of mirrors, and making adhesives. Molasses, an important byproduct of the cane sugar industry, widely used as a livestock feed for making alcohol drinks such as rum and for the manufacture of host of chemicals. Molasses can be used as a minor component of mortar for brick work. Ink rollers on printing presses were originally cast using a mixture of molasses and glue. Molasses can be added to the soil of almost any plant to promote microbial activity. Sugar bagasse, a fibrous portion of the sugar cane that remains after the juice has been removed, is burnt as boiler fuel, but is also used in the manufacture of paper, insulating wall board, fireboard, cardboard, plastics, and furfural, the latter being important in oil refining and nylon industries. Cane wax after extraction with solvents is used in the manufacture of polishes, cosmetics, and paper coating. Example, carbon paper. The filter cake is used as a fertilizer because of its high calcium, nitrogen, and phosphorus content. Thick noble canes, which are relatively soft with a high juice and sugar content and low in fiber are chewed throughout most of the tropics. So, I conclude today's lecture with these remarks that the sugarcane needs a hot and moist climate. The optimum average temperature for the growth is 25 to 26 degrees centigrade. The water requirement is high. In most growing areas, the cane needs 1500 to 1800 millimeters of rain. I hope you might have understood this lecture